This very quick video will cover the last part that you need to know about the formation of ATP and the formation of energy through glycolysis and aerobic respiration and whatnot. So, to sum up everything, we have glycolysis, form pyruvate in the cell cytosol. Pyruvate can either form lactic acid through anaerobic respiration and thus regenerate that NAD. Remember, it's very important of why we form lactic acid, really to regenerate NAD. Or it can go into the mitochondria, which I probably shouldn't put it there, to do the Krebs cycle. And you form all that NAD. Remember, each cycle of the Krebs cycle forms 3 NADH, 1 FADH, and 1 GTP. Now, the process of just forming pyruvates to acetylcholine, which is the first substance, forms 1 NADH by itself. It happens twice, so you form 2 NADH. Probably should. In this process, this cycle happens twice, so you form 6 NADH, 2 FADH, 1 GTP. All that stuff goes to the electron transport chain. The electron transport chain takes all these electrons out of the NADH, FADH, and takes the energy from these electrons and forms ATP by combining with water and oxygen. And thus you add these all up. This is 30 ATP. But then you have, this all is 30 ATP. In this process of glucose to pyruvate also forms 2 NADH. And that NADH is why we're forming lactic acid in order to reform NAD to keep that cycle going. It also forms 2 ATP this whole cycle. It forms 4 ATP, but you lose 2 ATP by doing this cycle, so you get a net of 2 ATP. That's why when I say when you do fast glycolysis or anaerobic, you do get 2 ATP. The process of forming pyruvate to lactic doesn't get you 2 ATP, but you get 2 ATP from the cycle. So you got 2 ATP. You add these all together, you got 32 ATP. Remember, these are 3 ATP each. You got 38 ATP. Now, you might see different numbers like 36 ATP. Remember, 39 is as if you use glycogens. You don't have that -1. But you might see 36 ATP also. You're probably wondering, how do you get 36 ATP? The math leads up to 38 ATP. Now, these 2 NADHs are in the mitochondria. The mitochondria is where the electron transport chain happens, which is where these NADHs get converted to ATP. So you got to get these NADHs into the mitochondria. How do you do that? Unfortunately, the mitochondrial membrane is impermeable to NAD. So pretty much what happens is you have your mitochondrial membrane. NADH is chilling out here. It's trying to get in there, and the mitochondria is like, fuck you, get out of here, and doesn't let it in. So we have to have some way to get the NADH into the cell's mitochondrial membrane. So very briefly, I don't know if she'll go over it. Moffat went over it briefly. You have something called the shuttle systems. The shuttle systems are just a fancy way of the body to pretty much play hot potato and take that hydrogen from that whatever, because the hydrogen is really what we want. The hydrogen has the electrons. That's what we need to get into the electron transport chain. So one shuttle system takes this hydrogen, brings it into the mitochondrial membrane, and converts it to an FADH instead of an NADH, and thus you got your hydrogen. Now remember, FADH gives you less energy than an NADH, so that's why you get 36 ATP. You can do all the math. If NADH went in there, you would get 6 ATP because remember you have 2 of them. But since you're shuttling only the hydrogens and you're shuttling them into it and then becoming an FADH, you're only getting 4 ATP because remember FADH is equal to 2. So you do the math, it gives you 36 ATP. That's one shuttle system. Another shuttle system is called the maltate-aspartate shuttle system. Well, this is called the glycolaldehyde 3-phosphate shuttle system. The other shuttle system is called the maltate-aspartate shuttle system. That's taking NADH and essentially bringing it into the mitochondrial membrane. If you bring it into the mitochondrial membrane, it forms a 438 ATP. That's called the maltate-aspartate shuttle system. I'm not sure how much you went over this. That's why I'm not going to waste a 10-minute video going over this system. but you have these two shell systems that brings in that residual 2 ATP or 2 NADH that you form from glycolysis to get into the mitochondria in order to act in the electron transport chain. So two shell systems, glyceraldehyde 3-phosphate shell system, maltate shell system, maltate aspartate shell system. Very simple. So now you know everything that you need to know about ATP and glycolysis. So now we'll very quickly go over a fun question of how do we form NADH, NAD from the electron transport shell system? From the whole process of glycolysis. Now, this is memorizing the steps. Now, you reform NADH from the process of going from pyruvate to lactate. This is just a fun question to go over to think about. But these are the type of questions you have to think about. The process of going from pyruvate to lactate reforms an NAD. You also reform an NAD if you use one of the shell systems. Because what happens in the shell system is the NADH, remember, bring the NADH into the cell's mitochondria. NADH can't cross the mitochondrial membrane. So what happens is you take this H and you bring the H into the mitochondrial membrane, and thus you reform the NAD. So those are two ways you can reform NAD. So just think outside the box, think critical of how you reform NAD through different processes.